In the annals of scientific discovery, the year 2018 marked a pivotal moment, the birth of Zhongzhong and Huahua, two identical long-tailed macaques. These primates, born 8 weeks apart, were not conceived through the natural course of reproduction. They were the product of a groundbreaking feat, successful cloning using the Somatic cell nuclear transfer, or SCNT technique. This achievement, spearheaded by a team of Chinese scientists at the Chinese Academy of Sciences in Shanghai, shattered long-held scientific barriers and ushered in a new era in the realm of genetic engineering. The implications of this breakthrough resonated far beyond the confines of scientific journals. It ignited a global conversation about the very essence of life and the ethical boundaries of scientific exploration. The success of Zhongzhong and Huahua's cloning was not just a triumph of technical prowess. It was a testament to the relentless pursuit of knowledge, years of dedicated research, and the unwavering belief in the power of science to push the boundaries of the possible. This achievement opened up a Pandora's box of opportunities and challenges, forcing the world to confront the profound implications of human cloning, a prospect that had long been relegated to the realm of science fiction. At the heart of this scientific triumph lies a technique known as somatic cell nuclear transfer, or SCNT. This intricate procedure involves the delicate manipulation of cellular components, essentially reprogramming a cell's destiny to create a genetically identical copy of the donor organism. Imagine a cell, any cell in your body, a skin cell, a muscle cell, or even a hair follicle. Each of these cells, termed somatic cells, contains within its nucleus the complete genetic blueprint of the organism. SCNT harnesses this fundamental principle of biology. The process begins with the extraction of the nucleus from a somatic cell of the donor organism. This nucleus, carrying the donor's complete DNA, is then transferred into an egg cell from which the nucleus has been removed. The egg cell, now endowed with the donor's genetic material, is then stimulated to initiate cell division and embryonic development, effectively transforming it into a cloned embryo. This embryo is then implanted into a surrogate mother, where it develops gestationally until birth. The successful cloning of Zhongzhong and Huahua was not a stroke of luck. It was the culmination of decades of painstaking research and the overcoming of significant scientific hurdles. Primate cloning, in particular, had long presented a formidable challenge, primarily due to the intricate complexity of primate egg cells. For many years, scientists had struggled to adapt the SCNT technique, successfully used to clone Dolly the sheep in 1996 to primates. The intricacies of primate egg cells, particularly their sensitivity to manipulation and the unique molecular mechanisms governing their development, had posed significant roadblocks. Early attempts to clone primates often resulted in developmental abnormalities or embryonic failure. The delicate dance of cellular reprogramming, essential for SCNT to be successful, proved particularly challenging in primate cells. The nucleus of a somatic cell, accustomed to its specific role within the body, needed to be coaxed into behaving like the nucleus of a newly fertilized egg. A feat easier said than done. The breakthrough in primate cloning came with a deeper understanding of epigenetics, the study of heritable changes in gene expression that do not involve alterations to the underlying DNA sequence. It turned out that the DNA in a somatic cell carries epigenetic markers that influence gene expression patterns specific to that cell type. These epigenetic markers, while essential for normal cellular function, were found to interfere with the reprogramming process during SCNT. The Chinese scientists, recognizing this obstacle, pioneered the use of epigenetic modulators molecules that could effectively erase these epigenetic marks and reset the somatic cell nucleus to a more embryonic-like state. This innovative approach proved to be the missing piece of the puzzle. By reprogramming the epigenetic landscape of the somatic cell nucleus, scientists were able to overcome the developmental hurdles that had long plagued primate cloning. This breakthrough paved the way for the successful birth of Zhongzhong and Huahua, ushering in a new era of possibilities in the field of genetic engineering. The successful cloning of primates ignited a firestorm of ethical debates worldwide. The prospect of replicating this scientific feat with humans, once confined to the realm of science fiction, suddenly seemed within reach, raising profound moral and societal questions. At the forefront of these concerns is the sanctity of human life and the potential for its commodification. Critics argue that human cloning could erode the very essence of what it means to be human, reducing individuals to mere copies of a genetic template. The potential for psychological harm to cloned individuals grappling with issues of identity and purpose adds another layer of ethical complexity. Furthermore, the possibility of designer babies tailored to possess specific traits raises alarms about eugenics and the potential for exacerbating existing social inequalities. The spectre of a society where individuals are valued based on their genetic makeup rather than their inherent worth looms large in the ethical debate surrounding human cloning. Beyond the ethical dilemmas, the potential psychological impact on cloned individuals remains a significant concern. Growing up knowing they are genetically identical to another individual could have profound implications for their sense of self, individuality, and purpose in life. The pressure to live up to the expectations set by the donor's life experiences, or conversely the fear of being perceived as a mere shadow of the original, could weigh heavily on the psyche of a cloned individual. The lack of a unique genetic heritage, a cornerstone of personal identity, may also contribute to feelings of rootlessness or a diminished sense of self. Moreover, the social stigma associated with being different or unnatural could lead to discrimination and social isolation. The psychological well-being of cloned individuals, shaped by a complex interplay of genetic inheritance and social conditioning, remains an uncharted territory fraught with ethical considerations. The advent of primate cloning and the looming possibility of human cloning have spurred governments and international organizations worldwide to grapple with the ethical and legal implications of this technology. The result is a complex and often conflicting tapestry of regulations governing human cloning research and applications. In 2005, the United Nations adopted a declaration prohibiting all forms of human cloning that are incompatible with human dignity and the protection of human life. This declaration, while not legally binding, reflects a broad international consensus against reproductive cloning, the creation of genetically identical human beings. Individual countries have enacted their own laws and guidelines, reflecting varying ethical and societal perspectives. Some nations, like the United Kingdom, permit therapeutic cloning, the creation of cloned embryos for research purposes, particularly for deriving stem cells for regenerative medicine, under strict regulatory oversight. Others, like the United States, lack comprehensive federal legislation on human cloning, resulting in a patchwork of state laws and ongoing ethical debates.